Kia ora, and welcome to this Manaaki Whenua LINKOnline Webinar. Today we're talking about trees again and sharing research that has some very practical implications for restoration practices in New Zealand. Peter Heenan is a botanist with a special interest in biogeography and phylogeny. His research includes studies of kānuka, manuka and kowhai, alpine plant biodiversity, and the conservation of unique limestone flora. I'll be back at the end to help with your questions, so don't forget to put them in the chat box as soon as you can. Now over to you, Peter.
Great, thank you, Christine. Tiffany. Good morning, everybody. Thank you for joining this webinar. So we'll just get started on it. In a minute, let me just move my picture of myself off my screen. So this is on ecosourcing, and it's the work I'm presenting here is based. Sorry, I'm just having trouble getting my keyboard to work. Is based on a paper we published about 3 months ago. It's open access, so it's freely available on the New Zealand Journal of Botany website. The authors are mostly a team of people from Landcare Research, but we were joined by Matt Lacombe, who's a professor at Otago University. And it's a pretty diverse team with specialist skills in genetics, ecology, and I guess myself with plant systematics. The outline of this presentation today is I'll first of all go over a little bit of the background to ecosourcing and some of the historical aspects of it and where we ended up today with it. We've got 5 issues that I just broadly cover. And then I guess the most interesting part is what does the science say? And so to present this, we did in the paper a bit of a literature review on 5 aspects of the science. And then we conclude with our what we call our eco-evolutionary approach, where we propose 9 rather large regions for New Zealand. So in 1972, Eric Godley, who was director of DSIR Botany Division at the time, wrote this short article in Forest and Bird, in their natural state, does planting achieve its purpose? And there's some important features of this article. The first is that it's the title, in their natural state. And throughout the article, Eric was referring to areas of indigenous vegetation that were in parks, national parks and reserves, for example, where people or organisations were planting native plants that weren't necessarily from that area. And that was Eric's primary concern, was the mixing of non-natives into these existing remnants of or intact pieces of indigenous vegetation. And in this article, he suggested that the source of plants should be no more than about 3 miles away. So this actually is quite a different approach to how we look at ecosourcing today, where a lot of the restoration work is in severely degraded habitats or where there's no native vegetation at all, and we're looking to restore what was there previously. There is still work going on with around established pieces of bush, but primarily a lot of the restoration work is restoring what has been missing. So there's quite a subtle difference in the approach there. Anyway, moving right on to the present day, ecosourcing today is well established in, for example, regional council policy statements, where they argue the closer the seed source to the restoration project, the better, or in the case of Taranaki, using locally eco-sourced plants to prevent genetic contamination of Taranaki stock, whatever that may mean. Anyway, at a Tane's Tree Trust conference in 2009, the question was asked amongst the 80 attendees or so at that conference, are current eco-sourcing policies and practice scientifically valid? 92% of the respondents said no or they don't know. Only 8 people said yes. So for a long time now, there's been some very clear uncertainty around what an eco-sourcing seed zone might look like. So how have we approached the topic? Well, we've got these 5 issues that I'll broadly summarise next, and then we look at 5 lines of evidence that we present from the science. So the first issue is we have these irrelevant conservation objectives for dynamic environments. So what do we mean by that? Well, I guess it's the second point here is the main one. Creating copies of local ecosystems of yesterday and today is considerable a desirable outcome. So we're looking to replicate what is already there. But one of the problems here is that we have these local extinctions, and there can be, and this is through the disturbed landscape that we live in today, there can be missing functional groups, missing species, and missing successional stages. And these are often not considered. We go to a nearby piece of bush where we want to collect our seed from, and we assume that that is representative of yesterday and today and where we need to collect our seed from. So in this regard, there's no consideration really of future states. And that leads to the second point. What are these future states? And the two main, the main point here really is we should be considering, the authors of the paper and myself and my colleagues consider the climate change and human modified habitats are not often considered with local ecosourcing. We're focused very much on the past and the present, not how climate change should be considered for the future, and that's particularly with regard to warmer winters, fewer frosts, wetter western and dry and eastern, etc., with the steepened rainfall gradients. Distributional ranges are interesting. We're on the sort of, I guess, well past the end of the last glacial maximum, but Peter Wardle estimated that less than 25% of our indigenous plants occupy their potential range. So in actual fact, if we consider moving native plants around the landscape, this range expansion is not all negative. It doesn't need to be done willy-nilly. It needs to be done with careful thought, but we're in that position whereby now we live in this highly modified environment, but many of the species aren't really at their distributional optimum. Also, in this modified environment, we have many disturbed and human modified habitats, and these can facilitate the establishment of native species well outside their typical habitat. And so not only we're talking about range, distribution being extended, but we're talking about plants growing in modified or different habitats. And there's the issue related to this about naturalizing natives outside their current range. And we have lots of good examples of that with Pittosporum, crassifolium, and Coprosma repens in coastal habitats. We also have these novel ecosystems today, and we have these guilds of carnivorous mammals, and we've got nitrogen-fixing shrubs. And fire is a major issue and has been for a long, long time. And George Perry and colleagues summed it up. Fire is we may have shifted large areas into successional traps from which escape from fire-prone successional communities to more resistant tall forest is difficult. So if we look at succession from early, mid, and late succession or climax communities, we are struggling to get the resistant tall forest from the climax communities in place. And the fifth issue is around threatened species management. And we probably should be considering moving rare plants to places where the risks to them are reduced. And it might well be that some of these sites are well outside their natural range. And we also need to consider the very small population size of many of our threatened species. And if we can mix them up, we can increase mating options, plant numbers, and to some degree, genetic diversity. And we already do this with animals. We've got many examples with birds, for example, of moving them well outside the historic ranges. So that begs the question, why not plants? So what does the science say? Well, the first bit of evidence we presented in the paper was around forest clearance and remnants. We've had extensive deforestation of the lowland forests in particular, and we've now ended up with about 30% of pre-human forests. We have the threatened land environments, which I'll show the figure very shortly, but we clearly have large areas, particularly of lowland New Zealand, where there's less than 10% of the original vegetation remaining, and it's got very low protection. And that also coincides with what we have as a depleted dry land zone. And the consequences of this forest clearing is we've lost connectivity and we have many small, isolated, disturbed, unrepresentative and unplanned remnants, especially in the coastal lowland zone. So this is a commonly presented set of figures where we can have the pre-human, the Maori period of settlement, and then the post-European settlement maps of New Zealand showing the clearing of principally our lowland forests. And this has created many small fragments over very large areas where there is often very little indigenous forest vegetation remaining. And of course, we have the threatened land environments, of which there's been 2 iterations. The most recent one in 2015 is shown here. And we can see, like the red, for example, is category 1, less than 10% of the cover left, and it's very poorly protected in those zones. Interestingly, if we look at forest composition throughout New Zealand, and this figure here is from a Matt McGlone paper looking at conifer abundance in samples that date back 20,000 years, we can see how dominant conifers were. And I just want to briefly pick up on that theme for a second, because the conifers really don't come in until late successional stages when they form a climax community. But in many of our lowland areas, we're not in that present state, and the conifers are fundamentally missing from much of our lowland remnant vegetation. They do occur in some regions, we're not saying they don't, but generally speaking across New Zealand, there are very low numbers, and as a consequence, they're poorly represented in restoration plantings. So it begs the question, how will those restoration plantings ever get to really a climax community, perhaps dominated by conifers, if we can't enlarge the seed collecting zones? So the second piece of science is around genetic variation and phylogeography. And if we just take a step back and think about the concept of local ecosourcing at the geographic scales that have been applied, that assumes, if we take a genetic perspective, that there has been extensive fine-scale genetic variation amongst our native plants. Well, in the paper, we did a review of 30 or more population genetic studies, most of just trees and shrubs. There's other studies on herbs, for example, and grasses, but we didn't include those. And of the 30 or so studies of the trees and shrubs, they generally have very widespread distributions of their genetic data. And in general, they don't support this notion of a very high level of fine-scale genetic variation. In fact, if you were to look at the genetic variation and you look at, say, the ecological regions and districts of New Zealand, where there's 85 ecological regions that people often use as a surrogate for defining a seed collecting zone, we don't get anywhere near even 85 regions of genetic variation for any of these plant groups. In fact, most of the genetic variation is often uniform across the country, frequently shows a north to south pattern, or there may be as few as 2 to 5 regional variants. So these landscape scale phylogeographic patterns are also consistent with major biogeographic regions, and there's an important aspect of that. The biogeographic regions are defined by multiple species sharing that landscape scale space, but the phylogeographic studies are of single species, and yet there's a lot of commonality between those 2 study areas. One thing that did come out, about a third of the samples had Cook Strait as an important phylogeographic boundary, but there were also 2 species, and they are Pseudopanax ferox and Pittosporum obcordatums, and their studies showed that those 2 species do have widespread distributions, but they have disjunct local populations that are genetically discrete. So they tend to go against the more general widespread trend. So what are 2 examples of what I'm talking about? So recent work that I've been involved with has been on Manuka leptospermum and Carnuka kunzea. And you can see here we have a very clear north to south trend in both of these species. The population structure of these is very strongly geographic, and there is really no taxonomic signal in there at all. It's purely a north to south with regional geographic variation. So the biogeography, this goes back to Cocaine's early work right through to the present day, and we've converged on having major biogeographic boundaries, of which there's about 2 in the South Island and probably 2 in the North Island, which defines 5 or 6 regions. We also have these smaller regions of endemism that have been defined by plants that are very restricted to those areas. But within those areas, they also have the common, more widespread species as well. So there's an interesting situation there around plant distributions and endemism. And 2 areas where that's occurring are in the upper, very northern part of New Zealand, and also in the Nelson region, for example. So for ecosourcing, large biogeographic regions can be recognised that share common biodiversity, ecological, environmental, and evolutionary history. And just 2 examples of the biogeographic boundaries, and the earlier work of Peter Wardle's, which was pretty much confirmed with our own studies more recently, where we analysed over 200,000 herbarium records looking at spatial distribution of richness and endemism, etc. And one of the things I guess we were able to do in that study was define more explicitly the South Island boundaries that we showed here are probably strongly related to the extent of the glacial ice sheet at the last glacial maximum. Anyway, the point is the boundaries are quite well agreed upon, even if there's a bit of wiggle exactly where they occur. The 4th bit of science is inbreeding depression, and this is really quite important when small populations occur, and this is when self-seed is generated because there's limited number of plants with limited mate choice, and also often the loss of pollinators. And this can have negative effects on plant performance and may result in unhealthy, doomed plants with cryptic recruitment failure. And not all inbreeding depression can be obvious in the very early stages when plants are being grown at a nursery, for example, and you wouldn't realise it until much later. And so we advocate seed should be sourced from large and healthy populations, even if distant from the restoration site, to overcome potential inbreeding depression. There's very few studies of inbreeding depression in New Zealand. One of the best is Dave Kelly and Alistair Robinson's work that they led on Ko-Fi. And the key thing about this graph here is that when the seeds, they have a high inbreeding coefficient, which means there's a lot of inbreeding, but that drops away as plants die, and the plants that are left have a low inbreeding coefficient as adults. So the inbred plants have basically been lost into the system, and we have outcrosses or plants that are not really affected by inbreeding at all to a severe extent. There's been 2 other studies that have shown inbreeding depression, and both on smaller shrubs, one a Veronica or Hebe, and the other some of our own work on Alleria adenocarpa. In both of these cases, the inbreeding depression was really stumbled across. It wasn't something that was being sought out. It was part of crossing experiments for other work. And the final bit of science we need to consider, which is really at the moment, probably in the realm of much future research that needs to be done, is ecosourcing of species and genotypes adapted to future environmental states, whether that be wetter, drier, warmer, etc. And there's 2 aspects to this. First of all, we have New Zealand-wide environmental assessments to distinguish major ecoregions, and so that's like a biome scale type approach. And there's been quite a bit of modelling done by the ecologists, particularly Jake Overton and John Leithwick around that work. Also, Jeff Rogers and Nick Singers have done some work in that space as well. And the 2 of the key drivers are the mean temperature of the coldest quarter and annual precipitation. So we did our own bit of modelling based on those, and were able to identify this area with this figure that had 8 regions. And we use this as the basis of our 9 with a separation in Cook Strait. And it does broadly mirror the earlier work of the other authors. And so the final real important point about this environmental shifts in climate adjusted ecosourcing is that the ecotype, the niche, or the habitat matching is really important. And the seed eco-sourcing sites should be similar to the restoration site in terms of the specifics of the habitat. And a good example of that would be, for example, manuka growing in a range of sites that have around Lake Ellesmere, Te Waihora down here in Canterbury, for example. We have it growing on the margins of the lake in several places, but you can look up into the Banks Peninsula and it can grow on dry clay hillsides there. So you would want to be matching those particular habitats to your restoration site. So this brings us to our eco-evolutionary considerations for eco-sourcing. And so our approach is that we advocate with a major loss of forests, we need larger areas for seed source, and these may be quite distant from your restoration site. Obviously, to overcome inbreeding, depression, we need large, healthy populations for healthy progeny. The phylogeography, we have these very broad, widespread genotypes that tend to go from north to south. Biogeography, we have well-established boundaries, and we advocate the sort of environmental matching at two scales, the broader biomes, but within those, the more specific niche or habitat differentiation for ecotypes. So when we summarize all that down, for the phylogeography, there is actually quite a bit of congruence. We end up, this is the 30-odd studies represented in the phylogeography graph here, and there is quite a bit of wiggle in the central part of the North Island. That's where the northern forest trees, they seem to have quite a few different southern limits, which have been reflected in the different phylogeography studies, and also reflected in the biogeography studies. Cook Strait, you can see there, is well represented. And then we have our environmental map as well. Anyway, when we very subjectively bring all this together, we have identified these 9 regions, which broadly look like this. And I guess the point here is we're not advocating 85 in terms of the ecological regions that we advocate. We're talking about much larger areas. And the other really important thing here is that we don't want to be wedded to the boundaries, and I'll come back to that in a second. So just as the talk draws to a conclusion, we need these large areas for eco-sourcing, and we need to consider some of the things going back to the issues I mentioned. We have these novel ecosystems, and they require the strongest indigenous elements that can be most successful. We have threatened species needing to be moved around into secure sites, and we shouldn't be necessarily too precious about doing that work. We need to overcome these local extinctions where we've lost major groups of organisms or plants, such as the conifers. And there's nothing really wrong with the assisted expansion of distributional ranges, although in terms of natives naturalising outside their distributional range, there is caution there and clearly more work needed. So just to reiterate, it's the concept of large areas that is important. Don't be wedded to the boundaries as we've presented them. Don't stress near the boundaries. Those boundaries are quite flexible, and they really just define 9 large areas as opposed to this concept of local and 5 kilometres down the road or the next adjacent piece of bush that you might want to source your seeds from. Ecotypes and habitats are very important, and we do discuss that in the paper in relation to Kānuka as well as Mānuka. And be practical about it. We're not writing a Bible that has to be, or a prescription that has to be followed to the letter here. We believe this approach will be better, but not necessarily perfect. And for future research, there's an urgent need, I believe, for phylogeographic and genetic variation studies of some of our key restoration species, such as kahuhu and karamu, and there's many others that would fit that category as well. Something like Pittosporum tenuifolium is naturally quite variable throughout New Zealand, and so the 9 regions might be a step too far for that, but it would provide a framework for at least being a bit more generous in that space about where the seed would be collected from. Something like Caprosma robusta, which is really an early successional colonizer and quite widespread, may well have very little genetic variation, and that would be our prediction based on the suite of species that have been studied already. I think the ideal goal where we could end up here is that we have species-specific guidelines once we can understand their morphological and genetic variation much better. So, for example, we might say for Pittosporum tenuifolium, you need these 10 or 12 regions that reflect the genetics, or for Coprosma robusta, you know, there might be just 2 or 3 regions. But I think we could end up in a place where we can make species-specific recommendations. And ecotypic and phenotypic variation is important for climate change, which is what the previous sort of point refers to. And, you know, the impacts of weedy species need to be considered as well, and they're not necessarily all bad. We just don't really understand their role in succession, and some of them may be very good early successional species that, with the right planting composition, would be succeeded in a planting, but do a very good job early on. So that's the presentation, and I'd just like to reinforce that there's much more detail covered on these subject areas in the paper, which is open access. So thank you very much.
Thanks very much, Peter. And you won't be surprised at the number and complexity of some of the questions that we've got rolling in. So I want to let everybody know that we will come to those questions, but some of it will be in writing with a quick note back to you later, and the Q&A is posted up on our website alongside the presentation in due course, once Peter's had a chance to look into some of those questions, because there's some pretty good ones in there. I'm just going to ask a couple of hopefully reasonably quick ones and universal ones. The eco-sourcing concept also includes collecting seed across a large range of individuals within the source population, obviously to get a broader and more representative genetic base. Can you comment on that?
Yep, so that's true. And if you look at the overseas literature, and actually the proposal here we're putting up is reflecting where the international literature has gone to with these seed collecting zones as well in terms of large areas. But the recommendations, which we also have in the paper, we adopt or promote one paper in particular, which suggested, you know, 50 seed collected from 50 individuals within a population, and obviously traversing that population, not collecting seed from within, you know, those 50 plants within a very small area where they might be brothers and sisters or cousins. You want to get across the particular community to get unrelated individuals. So that's covered off in the paper as well.
Great. More reading to be done. How do your recommendations around eco-sourcing practice compare with the international standards, such as the Society for Ecological Restoration?
Yeah, so we're quite consistent with that. And if you look at the work that's being done in Australia, North America, and Europe, for some particular species, they're recommending quite large seed collecting zones now, because as per the example there of say, you know, the Pittosporum or the Coprosma, they've done the detailed genetic work, and they understand the genetic variation across the species distributional range, and so they are defining these quite large, in some cases, seed collecting zones. And so the whole approach is actually, it's nothing really novel. It is new for New Zealand, and has certainly created a lot of interest, that's for sure, in discussion, which is a healthy thing, but it is actually quite consistent with the international literature.
And that leads to this one final question I'm going to ask. What size of forest is considered a large population for New Zealand and our native species?
Well, it's a bit hard to define actually, Christine, but if you lived in the Canterbury Plains, you know, a hectare would be probably a good sized bit of remnant forest. But if you were in other areas, it could be, you know, 10 times, 20 times larger than that. It really is case specific, and that's one of the issues we also discuss in the paper, is about the sort of broader application of this needs to be, you know, taking account of the landscape scale variation that we're seeing in a particular geographic area, the climatic variables, coastal to inland, for example. You know, we acknowledge some of the areas we've defined are quite large. You'd be quite silly, for example, to collect seed from a coastal area to plant in a montane area, and vice versa. So there's still some practical elements that need to be considered in this, and it's so I guess in terms to answer your question, it depends on where you are.
And that seems to be often an answer to many of the challenges that we tackle here at Manaki Whenua. I'd like to say a big special thank you to Peter and to everyone who's joined us today. It's been great to have you on board. We will answer those other questions. Watch this space, they're fascinating. And check your follow-up email for a link to the open access paper that Peter has talked about. We'll be back again with another webinar next month. Goodbye for now.
Thank you.